2, egy. Good afternoon. This is a short and mostly popular overview of the basics of the genetics, including the classical genetics. In the genetics, we can consider that there are eras. The Mendelian era, the discovering of the chromosomes, partly influenced by the politics, the era of the biochemistry, bioinformatics, and the system biology approach. Whom do we believe in? Left side, you see Thomas Hunt Morgan, who was awarded the Nobel Prize in Physiology and Medicine in 1933 for his discoveries concerning the role played by the chromosome in heredity. And together with Weissman, they told that the gametes, the so-called germ plasma, is mostly independent from the somatic part of the cell. It means that the environmental changes cannot be inherited. And right side you see Trofim Denisovich Lysenko, who was the main player in Stalin's war on genetic science. He told that even the changes caused by the environment can be inherited. They dismissed the results of the classical genetics. They told that the genes are imaginary things, and scientists like Nikolai Vavilov was dismissed and at the end killed owing to this so-called science politics. The chromosomes are carriers of the inheritance, now we know, and we can make some morphological groups. This is a chromosome set of a human. You see that this is 22 times 22 plus XX. This is a female. But instead of the XX, you can have X and Y. And this is a human X and Y chromosome picture. The number of the chromosomes N in the gametes or 2N in the somatic cells are very different. In human, 23, in the mouse, 20, and in the rat, 21. In the fruit fly, 4, or in the dog, 39, in the cat, 19, in the yeast, 16. So they are very, very variable, and there is no direct relation between the stage of development of the animal and the number of the chromosomes, because small number of the chromosomes may carry a lot of DNA. It is important to speak a little bit about the two types of the cell division. For the somatic cell, we have the mitosis. when the two sets of chromosomes, so-called diploid set, during the division of the somatic cells will be consumed. So from a cell of 2n will be produced two cells of 2n and 2n. During the production of the sperm and egg cell, it means that during the gametogenesis, the cell division occurs as a meiosis. The normal double set of, in case of human, 46 chromosomes have to be halves to 23 to ensure that the resulting haploid gamete can join with another haploid gamete, the mother's egg cell and the father's sperm cell, to produce a diploid organism. Here I have to mention previously one of the Mendelian Rules prerequisite: the gametes are created by random segregation. So the Mendel's laws of inheritance has prerequisites. He was using clean lineage series, that is, the ancestor of the individuals used in the crossing experiments unvariable showed the threat in question during many generations. It means in details, in modern language, the prerequisites. Number 1, characters are unitary, that is, they are discrete, that is, purple versus white, tall versus dwarf. There is no medium-sized plant or light purple flower. Number 2, genetic characteristics have alternative forms, each inherited from one of the two parents. Today these are called alleles. Number 3, one allele is dominant over the other. The phenotype reflects the dominant allele. Number 4, gametes are created by random segregation. Heterozygotic individuals produce gametes with an equal frequency of the two alleles. Number 5, different traits have independent assortment. In modern terms, genes are unlinked. And the clean lineage series may be mentioned as inbred strains in the laboratory animal science. So the Mendelian rules are the uniformity, the segregation, the independent assortment, and the purity of the gametes. Purity of gametes means that heterozygotic individuals will produce gametes with an equal frequency of the two alleles with the one or the other genes. And here you see the monohybrid crossing of yellow rounded and green rounded peas. The F1 generation was self-fertilized. F1 generation, all individuals have the same genotype and same phenotype, expressing the dominant trait, the yellow. In the F2 generation, the phenotypes in the second generation show a 3:1 ratio. In the genotype, 25% are homozygous, with the dominant trait, 50% are heterozygous genetic carriers of the recessive trait, and 25% are homozygous with the recessive genetic trait and expressing the recessive character. And here you see the explanation of the monohybrid crossing with chromosomes. Mendel made also dihybrid crossing, crossed the rounded and shallow yellow pea with a wrinkled and green one. The same as previously, the self-fertilization of the F1 generation occur. In the F1 generation, all individuals have the same genotype and same phenotype, expressing the dominant traits, yellow and shallow, not green and wrinkled. And you see that in the F2 generation you see the segregation of the traits. And here you see the explanation of the dihybrid crossing with chromosomes. The traits are inherited independently so that new combinations can occur. Average number of ratio of phenotypes 9:3:3:1. And a nice example for dogs, Boston Terrier crossed Dachshund. You have homogeneous phenotype in the F1 and segregation in the F2. Now we can formulate the Mendelian rules, the definition of the Mendelian rules. The first is the law of dominance and uniformity, which means that some alleles are dominant, others are recessive. An organism with at least one dominant allele will display the effect of the dominant allele. The second is the law of the segregation. During the formation of gametes, the alleles for each gene segregate from each other. Therefore, each gamete carries only one allele for each gene. And the third law is the law of independent assortment, means that the genes of different traits segregate independently during the formation of the gametes. The gametes, the spermatozoon and the ovum or egg cells. In human, the 23 chromosomes, 2 and the 23rd, gives more than 8 million possible combinations. Here you see the wild type male and female Drosophila with red eyes and yellow-greenish color, and with a lot of sexual dimorphism in measure. There are mutations, for example, the white-eyed mutation or the vestigial rudimentary wing mutation or the black color mutation. As many pair of chromosomes, as many linkage group can be found. This is a new discovery that we have genetically linked traits. The crossing over gave a possibility for change of genes For example, during the crossing of B mutant black and vestigial individuals crossed with a white-colored normal winged Drosophilus, and along with the independent assortment, the crossing over increases the genetic diversity by producing novel genetic combinations like black with wing or wild without wing. But in the animal world, many genes have more than two alleles. Many traits, for example, the height, the litter size, depend on many genes, and this is the non-Mendelian inheritance. Traits dependent on multiple genes are called polygenic traits. The environment may interact more or less, and it can be expressed with the h on the second, the heritability number. What about the sex determination? The sex determination is very important in the animal husbandry, especially in the laboratory animal science. We have to consider the anogenital distance. In male, this is large, and in female, right side, this is short. It can be seen on these pictures, the same, but in the middle we can see also a plug, a vaginal plug. What is this plug, and when can be seen? The plugs are useful for obtaining timed mating. A plug is a hardened, and to know that we had a successful mating in the night, for example. A plug is a hardened semen from accessory prostate, mostly, blocking the vaginal orifice and remain in place for about 12 hours after mating. Plugs are detected by visual inspection or by probing gently with a sterile toothpick or blunt probe on a female immobilized. Mating is assumed to occur at the midpoint of the dark cycle, midnight under the 12-hour on-off cycle, starting at 6 a.m., and thus noon of the next day is half days of the gestation. On the picture you can see vaginal smear. We have to see the cytology. We have to consider in the picture the round, oval and spindle-shaped nucleated epithelial cells and or the anuclear epithelial cell in the estrus on the second. The vaginal smear analysis is the preferable technique for accurately determining the estrus phase in mice. This technique was first described in rats based on the correlation between the layers of the characteristic desquamated vaginal epithelium and the hormonal profile for each stage of the estrus cycle. And beside that, how do I tell if a mouse is in estrus? Female mice in estrus will be receptive to mating. By picking females in estrus, they have swelling of the lip of the vulva closest to the anus. The sex determination systems. In some species there are hermaphrodites. There are also some species that are only one sex due to parthenogenesis, the act of a female reproducing without fertilization. In some species, sex determination is genetic. Males and females have different alleles or even different genes that specify their sex morphology, sexual morphology. In animals, this is often accompanied by chromosomal differences, generally through combinations of XY, ZY, X0, Z0 chromosomes, or haplodiploidity. The sexual differentiation is generally triggered by a main gene, a sex locus, with a multitude of other genes following in a domino effect. In some cases, the environment, especially the environmental temperature, will determine the sex. For example, alligator. Genetics of sex determination can be classified in the Drosophila type, like mammals, amphibia, fish, Orthoptera, insect like grasshopper, locust, and Hymenoptera, insect like bees and wasps, and the nematodes too. Here you see in a visualized form the XX and XY crossing that the result of meiosis is one type of egg and two types of sperms. This is visualized in this picture too, and at the end the female to male ratio is 1:1. The second class of the genetic sex determination is the Abraxas type for birds, reptiles, and butterflies, Hymenoptera. This is the ZZ for the males. They have two equal chromosomes, and ZY, ZW for the females, where the W is a very small, close to the 0 chromosome. And here you see the crossing of Drosophila melanogaster. You see two versions of the X-linked inheritance. Left side, the red-eyed female, the wild-type female, is crossed with the mutant red-eye males, and right side up, a mutant white-eyed females XX crossed by the red-eyed male XY. This is a discovery of Thomas Hunt Morgan. The title was Sex-Limited Inheritance in Drosophila, published in Science 1910. So this is in details the sex-linked inheritance 1, when the wild-type female was crossed with the mutant males, and you see that no white-eyed females appeared. And the second version, when the white-eyed female was crossed with the red wild-type red-eyed males, this is the second version. We have to mention also the human hemophilia as an X-linked inheritance. And we arrive to the biochemistry of inheritance, DNA, and 40 years ago we had only these figures of the DNA and the two base pairs, the adenine with the thymine and the guanine with the cytosine. But today we have a very modern representation of DNA illustrating the three-dimensional structure of the double helix. Left we see the so-called ribbon diagram. The sugar phosphate backbones are depicted as bands with horizontal lines used to represent the base pairs adenine, thymine, and guanine, cytosine. And right up you see a computer model of the DNA molecules. The sticks figure are the sugar phosphate chain winding around outside the stacked base pairs, forming a major and a minor groove. How is the duplication? This is the original Watson-Crick model of DNA replication, which is a semi-conservative. In the daughter string, you have one from the maternal strand, and one is a new one. What are the events during the stages in gene expression in eukaryotic cells? First, there is a trigger which causes the chromatin modification in the nucleus. The DNA will be unpacked, involving histone acetylation and DNA demethylation. These are also part of the epigenetic inheritance. And after the unpacking DNA, the genes are available for transcription. The transcription occurs on the RNA. This is a primary transcript, and after the RNA processing, the ready messenger RNA will be transported to the cytoplasm. And in the cytoplasm, on the ribosomes, the translation, the protein synthesis occur. And according to the length of their half-time, the messenger RNA will be degraded. The polypeptide, the new polypeptide, after cleavage, chemical modification, will be transported to cellular destination, where the active protein is an enzyme or a structure protein. And of course, at the end, this protein will also degrade it. The starting point of the transcription is interesting. We need a starting point. This is a promoter of eukaryotic cell gene and cell, of course. And the promoter, many transcription factors can be binded on specific frequencies. You can see left on these pictures. The transcription factors in interaction with each others and with the RNA polymerase enzyme are helping the starting of the transcription. The gene expression is regulated by the transcription, by the messenger RNA processing, cutting out the unnecessary part. In the cytoplasma, the half-life, the messenger RNA stability, how long is it, and the protein synthesis of messenger RNA, ribosome, and the transfer RNA, that is, means that the process of the translation. The DNA-binded histone proteins fixed by zinc fingers may modify this process. And the epigenetic changes, the state of the DNA methylation and histone acetylation are also very important. These are part of the epigenetics. The post-genetics, after the classical genetics. Even Schrödinger in 1940 mentioned that the genetic information should be coded in aperiodic crystals without knowing about the DNA. And in case of the RNA, the interference and the presence of picnon, micro RNA and silencing RNA has been discovered. Pelionis wrote that the inner mathematics of the genome is a fractal geometry which assure a condensed information storage. I have to mention the fractals from my other lecture about the chaos and complexity. Fractals are peculiar mathematical formations with a high level of self-similarity. You see here the central dogma of the genetics, that there is only information from DNA to RNA, and there is no feedback from protein to DNA, and also that there is no information in the junk DNA. This central dogma proved to be false, and this double lock removal of the double lock allows the principles of recursive genome function. Some word about the RNA interference and the micro and small interfering siRNA. Here you see a visualization of the RNA interference, that the messenger RNA is destroyed before it is translated into protein. The RNA interference refers to a phenomenon where small pieces of RNA can shut down protein translation by binding to the messenger RNAs that code for those proteins. RNA interference is a natural process with a role in the regulation of protein synthesis and in immunity. In details, the small interfering RNA, siRNA, sometimes known as short interfering RNA or silencing RNA, is a class of double-stranded RNAs, at first non-coding RNA, typically 20-24 base pairs in length, similar to microRNAs, and operating within the RNA interference pathway. It interferes with the expression of specific genes with complementary nucleotide sequences by degrading messenger RNA after transcription, preventing transcription. Here you see the principles of recursion, iterative access to regulatory DNA. The growth of fractal Purkinje neurons of the cerebellum is governed by the aperiodical discrete quantum system of sequences of DNA bases, codons, and motifs. The full genome is fractal. The discrete quantum system of Picnon-like elements follows the Zipf-Mandelbrot parabolic fractal distribution curve. Pelionis, 2008-2013. Picnons are specific human primate-specific DNA motifs, at least 16 nucleotides long, that are repeated in blocks in intergenic and intronic regions of the genome and can be located in a new class of non-coding RNAs of variable length. Recent studies reported that Picnon deregulation could be involved in the carcinogenesis process, including colorectal cancer. Here you see the healthy state when there is, during the growth of the Purkinje cells, there is a green recursive into by structural proteins binding with DNA, and regulatory into for recursive iteration, proteins and microRNAs binding with DNA. And we have a pair-used pockets methylated. And here you see an example to the Mandelbrot fractal, which is characterized by a high level of self-similarity but not equality. So this is a statistic similarity. And here is the fractal approach to recursive iteration, which has been used to identify fractal fractal defects causing a cerebellar disease, the Friedreich spinocerebellar ataxia. In this case, as runs disrupting a fractal regulatory sequence. In the yellow field, you can see that the DNA methylation compromised by epigenomic, environmental, or hologenomic hereditary factors, and unregulated cancerous fractal growth results. And finally, we have to know that the animals and the human, together with the carrying microbes in the microbiome, are superorganisms. They are complex ecosystems of the bacterial, viral, fungi, etc. Microbiome in the gastrointestinal tract, in the lung, on the mucous membranes and the skin. The cells in the cells, there are compartmentalization. So this complex ecosystem is characterized by metabolic compartmentalization. Each chemical process has own place, own vesicle, own sector in the cell. The health is determined by endogenous, like genotype, epigenome, circadian rhythm, age, and exogenous, food, feed, drink, xenobiotics from the environment, as well as the microbiome everywhere. So it can be studied and approached only by system biology approach. And now, never more junk DNA. We have non-coding RNA in an important role. Most of the mouse genome corresponds to that of the human, but the differences are mostly in the fine regulation. Thank you very much for your precious attention.